The first thing we should do with any data set is to look at the distributions. As we see in this example, Northern Ireland does not seem to fit at all with the rest of the data. It is considered to be an extreme outlier. While we do not exclude every value that we dislike, there is some rationale for excluding outliers. However, when reporting our process of data analysis, we should be very honest with our audience about the procedures we used to identify and eliminate confounding outliers. Just because two variables are correlated does not mean that one caused the other. It could, but a correlation does not prove that. We could have misunderstood the causality. Perhaps we assumed that happiness causes better relationships when actually better relationships cause happiness. Or consider that a relationship is only partially causal. Wealth may cause happiness, but so many other variables also influence happiness. Correlation may not take into account confounding variables or a third causal variable. Both variables may increase over time naturally, such that their correlation is meaningless. Finally, the relationship we may have found may be due to coincidence. If we can show that one variable leads to another, in both the presence and absence of other possible causal factors, we have more reason to believe the relationship might be causal. Randomization helps support the case for causality, especially with random assignment. Random assignment makes systemic differences between groups unlikely. Finally, if we can explain why we found what we did, the best thing to say is that a relationship is worth exploring further. The fact that a sample correlation coefficient is not exactly zero does not necessarily mean those variables are truly correlated in the population. I previously gave you some idea of how to determine the strength of a relationship, but we are also concerned with whether the relationship is significant. This not only has to do with the correlation that we find, but the sample size that we drew from. A student's t-test can help us calculate the significance. Notice that the formula represented in the text box to the right clearly shows that degrees of freedom should be included, whereas the formula was slightly different on the bottom left. The degrees of freedom represented in the divisor. After calculating the t-value and taking into account the degrees of freedom, we can calculate the probability based on whether our test was one-tailed or two-tailed. Both results are significant at the 0.05 level. Remember when I said that degrees of freedom matter as does the sample size? Take a closer look and realize that degrees of freedom is actually calculated from the sample size. A significance test tells us whether or not we have reason to believe that the true correlation in the population between two variables is different from zero. However, it doesn't tell us very much about what the true correlation in the population actually is. For that, we want a confidence interval. We only have a confidence interval if we can state a probability of about 95% that our value falls within that interval. A statistic that falls within a confidence interval is still at risk of being wrong about 5% of the time. For this class, we're not going to calculate confidence intervals, but they can be done by hand or with the statistics programs. While we often do look at the relationship between only two variables, sometimes we want to understand the whole grouping of variables and all the relationships between. An intercorrelation matrix is a matrix in which each cell contains the correlation and related information between the variables on the rows and columns. In this slide, you'll see also a scatterplot matrix, which is a graphical representation of the relationships between all the variables. When we had one variable measured on a continuous scale, one variable measured as a dichotomy, then the correlation coefficient that we produce is called the point biserial correlation. Again, when one variable is measured on a continuous scale and another is measured as a dichotomy, then the correlation coefficient we produce is called a point biserial correlation. Yet another correlation coefficient is phi, Phi. It arises when both variables are measured as dichotomies. So if you are looking at gender and regular church attendance as a yes or a no, then both variables would be dichotomous, having only two answers. You can start to see how the characteristics of the data start to influence the type of statistical test that we will do. It may still be similar, but each test is adjusted for a specific type of variable and data. Effect sizes are the square of the correlation coefficient. If we take the correlation coefficient that we have calculated and we square it, the result can be interpreted as the percentage of variation in heart disease or whatever we are studying that we could predict, just knowing the other thing, in this case, pace of life in the city. In other words, they are squared in that example. It is the percentage of explanation in heart disease predicted by the city's pace of life. Since the correlation is 0.365, the R-squared value is 0.133, or 13%. While these numbers may seem small, the magnitude size is somewhat subjective to the field of study. If I could get 1% closer to curing cancer, I would be famous. Again, R squared tells us the percentage of explanation in a dependent variable. By that I mean explained variation. Here's an example of how we would write up the study on course evaluations concisely but accurately. This speaks about the hypothesis we were testing, the way we collected data, the statistical results, and the conclusions that we could draw. Here's a short example. Before we start with Chapter 10, we'll take a look back at concepts from previous chapters: an independent variable, dependent variable, a scatter plot, correlation coefficient, a regression line, standard error, and standardization. Before we get started, we will do preliminary screening and tidying, which I'll speak about shortly.